today I will be presenting my replication of the EPHOD experiment. EPHOD stands for Enzyme pH Optimum Prediction with Deep Learning, and it is a machine learning approach for predicting the optimum pH at which enzymes function. So let's get started here. So for the introduction part, we know that enzyme activity is highly sensitive to pH conditions, and hence predicting the optimum pH is very important for research and industrial purposes. And the EPHOD model uses protein language model embeddings to predict the optimum pH directly from sequence data, which is a significant step forward in computational enzyme discovery and design. So now let's talk about the methodology. For the dataset, they used a database called Brenda, which is Braunschweig enzyme database. They used 9,800 enzymes approximately with experimentally measured optimum pH values. The sequence were represented in several ways. One was encoding amino acid composition, engineered features, and embeddings from advanced protein language models, which are shown in the bracket here, given here, like ESM1B, ESM1B, PROT5. Now, the researchers also evaluated a wide range of machine learning models, including traditional models such as ridge regression, SVR, which is your support vector regression, random forest, XGBoost, then KNR is your K nearest neighbors, as well as they used also the neural networks like uh, FNN, which is your feed forward neural network, then CNN, DCNN is your convolutional, direct convolutional, RNN, RNN is your recurrent neural network, and then these AT ones, the LAT, RLAT, are those attention based architectures. Also, the training involved extensive hyperparameter optimization in where loss was related to address level imbalance and transfer learning using a large data set of environmental pH values for secreted bacterial proteins. The models were evaluated on a held out test set of 1971 sequences with a subset of 999 sequences having less than 20% identity to the training set for robust generalization. So now let's talk about the key results for this paper. As we can see in the given diagram, here the ESM1G ensemble, which is the model in top with the EPHOD, it was the top performing model as it is shown in the top of the chart here. So the best performing model was indeed EPHOD, which is an ensemble combining a RLAT, which is your residual light attention neural network, and a SVR model, the support vector regression, and they both used ESM1V embeddings on this. On the test set, EPHOD achieved the root mean square error of 1.25 pH units, and the R square value for these models were, as you can see, it is between 0.64 and 0.66, but the main point of focus here is the 0.661, which is for the ensemble or the EPHOD1, which is the model in top here. About the robustness, the model maintained high accuracy even on sequences with low similarity to the training data, with only a minor increase in root mean square error of only 0.12 pH units. EPHOD outperformed structure-based and biophysical methods in predicting the optimum pH. Attention analysis showed that the model focuses on residues near the catalytic center and on the protein surface, especially charged residues relevant to pH adaptation. Now talking about my replication, as we can see in the comparison given over here, the left side shows the original research predictions for the optimum pH values, As we can see, it was tried on 1971 sequences, while my replication was only on 250 sequences due to some computational constraints. So uh, the leftmost column shows the uniprot ID of the proteins and the enzymes, and the columns beside the rest of the three columns besides it are for those machine learning models which predicts the optimum pH. As you can see, the RLAT, the residual light tension, the support vector regression, and then finally the EPHOD, the ensemble one. And if you notice, the ensemble one is roughly the average between the RLAT and the SVR one. So that's a thing to keep in mind. So yeah, now I also made a scatter plot uh, for the values obtained for my replication and for the original research work published. So the original one was plotted on the x-axis, and the my replication was plotted on the y-axis. And as you can see, it lies on a straight line y equals to x. So it means that my replication was indeed really successful, because I was getting the same prediction values of the optimum pH for the same proteins. So yeah, so uh, hence my prediction were similar as compared to the paper, and it was similar in range and distribution to those in the original paper. The main challenge I faced was computational, which restricted the size of my test set. It was because the HPC I was using, it was using CPU instead of GPU to compute due to some I think PyTorch module error. I tried to fix it, but I couldn't in time, and the total time to test all the 1971 sequences was coming out to be around 30 or 33 hours. So it wasn't feasible for that mode. So I just decided to do it on a smaller data. So yeah, so in summary, my replication closely followed the methodology of the original research work, and my results are broadly consistent with the published findings. So now the future work, it could focus on optimizing model hyperparameters, increasing dataset diversity, or exploring alternative protein language model embeddings for further improvements. So that was it for my side. Thank you very much.